The potential safety hazards in cryo-EM are of many types. There's biological hazards, chemical hazards, and some physical hazards that we need to pay attention to. The first one that I'd like to talk about is the biological hazards. A lot of cryo-EM work is being done on human pathogens, often viruses or bacteria or other forms of life that are infectious to humans. And so obviously these pose biological risks. And when you're working with a pathogen, there's different approaches to minimize that risk. One of the approaches is, if possible, you can use light chemical fixation to debilitate the infectious agents and make them safe to work with. If you don't want to try light fixation, then another route is to use attenuated strains that are missing key genes or have some other deficiency which renders them much less infective than a wild-type virulent strain. If, however, you can't use an attenuated strain, or if the attenuated strain itself poses any hazard at all, then you need to take further precautions to protect yourself during a cryo-EM experiment. Now, depending on the risk that your sample poses, there are different levels of protection that you can employ. And obviously, you should consider these carefully and talk to your biosafety officer and make sure the procedures that you're planning to use are approved and adequate to protect you and others in the lab. The lowest level of protection starts with simply wearing the right clothing. So anytime you're working with a hazardous agent, you should wear a lab coat like I'm showing, and you should wear long pants, and closed-toe shoes are also important in the cryo-EM lab because we use a lot of cryogens that often spill on the floor, and so you don't want to be freezing your toes. In addition to this clothing, another layer of protection that can be added is the use of gloves. And so you may use a single pair of gloves or even a double layer of gloves as you feel necessary during your experiments. And anytime you're working with liquid ethane, you should wear safety glasses to protect your eyes from ethane splashes. In some cases, you may even feel better wearing a full face mask like this. And as you can see, with the full face mask, nearly all the tissues of my body are covered and protected from splashing. This equipment helps protect you from the hazardous agent that you're using. You can also protect your equipment. For instance, in a Vitrobot, you can wipe down all the surfaces of the Vitrobot where the pipette might touch with a disinfectant. Also, in some labs, they'll cut circular pieces of tin foil to go behind the filter paper pads on the Vitrobot blotting pads to prevent the sample from leaking through the filter paper and contaminating the pads of the Vitrobot. Of course, an even higher level of protection is to put the entire Vitrobot device inside a biosafety cabinet. Now, this lab was designed to have the Vitrobot in the same room as the biosafety cabinet for situations where we might culture a mildly hazardous agent in the biosafety cabinet and then move a small amount of that around to the Vitrobot and plunge freeze it, all with the door closed to protect others. And for even more extreme cases, you can put the whole cryo-EM lab in a BSL protected facility, including the Vitrobots and the cryo-EMs. Dealing with a biologically hazardous agent in a cryo-EM experiment can be thought of in 3 stages. The first is culturing the agent until it is frozen. The second would be handling that agent after it's frozen. And the third would be discarding it after it was imaged. And it's important to remember when considering the hazards here that the degree of hazard is related to both the amount, the total amount, and the concentration of agent that you're working with. As you plan your experiment and describe it to a biosafety officer, you'll be informing them about how much total culture you plan to produce. And it's important to note that after that culture is produced, when you plunge freeze it on a grid, there can be a very small amount on the actual grid, fractions of a microliter. In fact, when working with, say, an infectious bacterium, after you plunge freeze a grid, there may be only several hundred cells on that grid. And so that's an important point to note as you consider the risks. That being said, the whole point of cryo-EM is to preserve those cells in as nearly native a state as you can. And it's been shown that many cells, after being plunge frozen, like is accomplished by the Vitrobot, if they are then allowed to thaw out in growth media, they will remain alive and infectious. So it's important to remember that while they're on the grid frozen, they may still be able to recover a living state, but their numbers are small. Now, it's in this context that I'd like to talk a little bit about puncture hazards. There are many different kinds of freezing devices. The Vitrobot is just one. And each different device has a different level of hazard of actually having a skin puncture. You notice that the tweezers that are used are very sharp. Some devices are much less automated. And in fact, some you actually take a piece of filter paper in your fingers and you touch it to the grid to blot off most of the sample before you plunge freeze it. And when your fingers are near that sharp tip of the tweezers, there have been cases, many cases, where people have plunged the tweezers and they puncture through any kind of glove the person is wearing and even can puncture the skin. And if you're working with an infectious agent, you need to be aware of that hazard and know how to minimize that risk and what you would do in the case of an accident like that. So the puncture hazard is at the moment of freezing. But remember, at all stages of handling your grid after it's frozen, into the grid boxes, into the storage devices, taking them back out, loading them into the electron microscope, and then taking them back out of the electron microscope and discarding them, that you're always handling these grids with sharp tweezers. And so protect yourself against puncture hazards at all times. Also remember that in addition to you using the equipment, you might understand the hazards of your sample very well, but remember that there are many other people who will use the lab and also the electron microscope, especially the service personnel. And so it's important to inform the service engineers of all the risks that they could potentially face in servicing a microscope where a grid, say, might have dropped into the column. Let's think for a minute about what would happen if a grid were dropped out into the column region. First of all, it would thaw out very quickly because the rest of the microscope is not held at liquid nitrogen temperatures. And once the grid thawed out, then any solution that was on it would violently boil away because of the extreme vacuum conditions within the column. So it's very likely many pathogens would be destroyed by thawing out in such a high vacuum. But nevertheless, maybe there are some viruses that would not be, or perhaps spores. And so you need to pay attention to all the possibilities. Once the imaging is done, most biological hazards are mitigated by simply discarding the grid into bleach or some other disinfectant. Finally, remember that after your cryo-EM experiment, you should clean the tools and disinfect them as necessary so that they pose no hazard at all to the people that will follow.